Okay, so some of you know me, some of you do not. I'm a faculty here in the biology department. I go by Dr. Y, but the full name is Yamada Hunter. So you can just, I like Dr. Y, it's cute. You just call me Dr. Y. It is my absolute pleasure to introduce a good friend, but also a really distinguished professor at UCSF, that's University of California, San Francisco, Dr. Saul Villeda. So we sort of overlapped because of our joint time at Stanford University, but Saul currently is an associate professor at UCSF. I'll tell you a little bit about his background. He went to UCLA, born in LA?
Oh, yeah.
Yeah, born and raised in LA, went to UCLA for his undergrad. He majored in physiological science, which is just a fancy word for biology. From UCLA, he went to Stanford University for his PhD and worked in the lab of Tony Weiss-Corey. And then from his PhD, this is where things get, I think, crazy interesting. So usually, if you want to become a faculty like Saul at UCSF, then you would have to do something called a postdoctoral degree. But Dr. Villeda is so incredible. He actually started a lab straight out of his PhD. And he was what's called a Sandler Fellow at UCSF. So without any postdoctoral experience, went straight in, started up a lab, and has been at UCSF ever since, became assistant professor and then associate professor. He's now the director of the Bakar Aging Institute. And I could like go on and on about all the awards he's won, but I'll just tell you a couple. In 2023, he got the Byers Award in Basic Science. In 2020, the McKnight Brain Research Foundation Innovator Award. He's also an endowed chair already, which is pretty cool, in biomedical sciences. He got that in 2019. So what Saul's going to tell us about today is really a couple overarching questions. He's interested in why the brain ages. I think this is something we're all intimately aware of because hopefully most of us have older parents or grandparents that we've interacted with. And sometimes you can see this, some of that cognitive function declines with age. So an overarching theme of his research is why the brain ages. And more importantly, can we reverse this aging? So can we help folks who develop things like dementia and have a really hard time recalling memories or doing daily functions? And I'll just give you the punchline, but I won't take too much away from Dr. Villeda's talk. He's been able to find that there's really interesting proteins and other factors in blood, especially the blood of young people or exercise-induced proteins, that when you give it to an aging individual, you're able to actually reverse some of that brain decline that we see. So this is the punchline. You can reverse aging, and Dr. Villeda has figured out some ways to do that. The last couple bits, this work has been so impactful that it's been featured in the New York Times. It's also been featured on NPR, which is super cool. Saul will tell you at the beginning, right, a little bit about his personal journey. His parents are actually from Guatemala. And one of my favorite quotes that I was creeping on you. One of my favorite quotes that I was able to pull out from Dr. Villeda was that I never pictured myself as a scientist until I was one. I think you'll tell us a little bit about why it was difficult to picture yourself as a scientist and the journey to becoming a scientist. Yes. Please join me in
Wow, that was crazy.
Before we start, can we make sure your slides work on the Zoom? Because do you have it shared right now?
Do have it shared.
Let me see.
Is it moving?
I wonder if you need to turn on your camera. He's not seeing it hasn't shown.
Oh, yeah, but he is sharing his screen.
I can try again.
Oh.
Share. This one. Share.
Oh, now it's
Now we're good.
That's working.
Yeah.
Okay, great.
I just did it again.
okay, great.
Everyone can see it?
Yes.
Perfect. All right.
First of all, hi, everybody. I'd love to thank Dr. Y for the invitation to get to meet all of you and share a little bit about the research that we're doing up at UCSF. And
Actually,
again, the whole idea, yeah.
sorry, we can see your whole presenter view.
Oh, no.
Sorry.
Um,
Sorry for the technical issue.
no problem.
What happened? Like,
Should we just leave it like that? Should we-
Yeah.
I think we could just leave it like that. Is that okay?
Not this is like the.
Yeah.
So give me 1 second. Let's stop sharing. See if I can see this.
Not computer, thanks everyone.
There's some room up here at the front to go on the floor. Sorry, there's not enough seats. Dr. White did too good a job without reading.
Do they still?
This is what it looks like, which is. We can see that.
What do they see?
Yep.
Yeah.
Is that okay?
We're good.
Oh, but then they can't see it.
Oh, no. Oh, yeah, then they can't see it.
They can?
And
No,
not
they
here.
can't see, but they can see.
But look at this.
Oh,
there
oh,
we go.
beautiful, beautiful.
We
Yeah,
got
that's
it.
work.
We got it.
Can they, what if I put it in slide view? Can they see it now?
Yeah.
No, then we see the presenter.
I can.
We're gonna
Let's leave
do-
it this way.
Okay, we'll
Okay.
go with that.
Does this work for everybody? Sorry.
Yeah,
I can make
we're
it
good.
Okay,
okay.
now we're presenter. I'm so sorry.
I don't, it shouldn't be though, right?
It's okay. I think what you'll do is just when you share screen, go to screen 2.
Not. Do you think if I do just the big slide view, it'll actually work now or be it?
Yeah.
Okay, fine. What do you see?
That works.
Ah, success, okay. Success. So Dr. Wise, whose class you want to take, by the way, she just solved this, so. And it's been a while since I've taught, so I'm obviously not going to be doing this anymore. All right, so first of all, I wanted to thank Dr. Wise for the invitation, as well as for fixing the slides and making it work. Again, that was a ridiculous introduction, and I remember sitting when I was an undergrad, and there were invited speakers, and you hear exactly what Dr. Wise just said, which is these accolades or what they've done in their profession, but you never find out what they're like personally or even how they got there. So I thought that I would share with you a little bit about why I do the type of research that I do, and that really has to do also with a lot of my trajectory. So I just called this brain-blood dynamics, and the mechanisms of brain aging and rejuvenation, and I'll explain what I mean by all of that. So the first thing is I'm fascinated by this idea of aging and cognitive decline, and we've all seen it at some point in time. We've met someone that's elderly and maybe they don't remember as well, or maybe you're talking about an experience and they'll go, oh yeah, that sounds really familiar, but they won't be able to tell you the details anymore. And to me, that's really intimately linked. That idea of cognition, of memory, is really intimately linked with your identity, who we are. We wake up every morning, maybe you have that chance to look at the mirror, and there's all these features that you recognize as your own. You know who you are. So think about it when you're driving, you're biking over. You have a sense of that identity, what makes you different from others. But that actually goes hand in hand. It's dependent on our cognitions. We're comprised of our experiences that we've had. And it's that sense of identity that I want to preserve. I really, education, science, a lot of that has been a really big part of my life and how I've gone to where I am. But that's also made me and given me part of my identity. And what I am sort of trying to avoid is as we get older, losing that sense of who you are. And that's something that's happening with natural aging, and that's something that's happening with age-related diseases, in particular things like Alzheimer's, right? You're losing that ability to even recognize yourself. You're losing your own identity. And the impact that that's going to have, not just on yourself, but on everyone around you. Everyone around you maybe sees you in one particular light, and as you lose yourself, you're impacting all those around you. And I think that there's ways that we can actually leverage biology to try and come up with therapeutics that may be able to slow down and potentially even reverse that. In essence, really maintaining who you are. And that's really what drives my research, is that big idea. Now, I've just made a really big point about identity. So how can I continue with the science without telling you a little bit about my own personal identity? And I'd like to share first my personal trajectory, a little bit about my scientific trajectory, and then my research trajectory. But notice that all of that is under this idea of identity. So for me, every experience that you have makes you who you are. So my personal identity is influenced by all of these. So a little bit about myself. This is a picture of me in the early 1980s. That was a long time ago. I had to scan this, and I mean, at least you can sort of see that I'm on a car there. It's getting old now. Like Dr. Wise said, I was born in California. I'm actually the first one born in California. My parents are officially immigrants, and I was the very first one born here. Here's a little picture of my family. That's my mom. That's another picture of me. My dad with another picture of me, and then my little sister who came around 11 years later. So there's a big separation between her and I. And again, we all sort of, I grew up in California. But my parents originally came from Guatemala, which is in Central America, and they came in the late 1970s. They actually came here illegally. So my parents were illegal immigrants. My mom eventually became a legal resident during amnesty. I, of course, was a citizen because I was born here. And then, later on, then my dad was able to become legal, and then that helped a lot. But that didn't happen until I was quite, late in elementary school, almost in middle school. So a lot of that early time actually happened in East LA. So I was born during the time of gangs, during the time of sort of a lot of drugs. So for example, where I grew up, I mean, it was very common to have drive-bys. That's sort of what led to my early personal identity, was really that kind of immigrant experience. We ended up moving to the high desert. Lancaster is sort of on the way to the Mojave Desert, still LA County though, as the edge of LA County. And there I went to high school. And then towards the end of high school, my dad really wanted me to become a mechanic. I have kind of skinny fingers, so you can really get in. And I thought that, that sounded good. They had the biggest house on the block. But along the way, I had always kind of hung out with my friends. So I had taken things like AP classes. I had done sports. I did anything to get out of my house because it was quite strict, like very immigrant strict family. But I found a loophole. Anything related to school was allowed, so I could just get out. So I did everything you can imagine. 6 o'clock, government, I'm there. Sleeping, but I'm there. So it ended up where I had a really good sort of packet at that time. I just didn't know it. So we were in a psychology class, and we were sort of telling the professor wanted us to pick the top 3 schools that we applied to and why we applied to it. And I had to go up to him off to the side and say, I didn't apply to college. Can I be excused? He freaked out. And mentors are so important because this awesome man went and saw the counselor, and they actually back then they called and everything, and there were a lot of schools that let me apply late. So I was able then to go to college. As part of that, even before college, but definitely once I got to college, there was this sense where I felt different because of sort of where my parents came from, how I grew up. All my friends, especially in classes that I were taking, did not look like me. A lot of them were quite affluent. So a lot of the experiences I had were just different. And I really like this image because I feel like it kind of shows a little bit about how I felt, and every once in a while I still feel it, which is kind of this idea of imposter syndrome, because I don't identify with those that are around me, I feel like I have to almost hide everything that makes me different. And if anything, that's actually what makes you stronger, makes you better. So I had quite a bit of imposter syndrome even before I got to college. Just that idea of I didn't apply and then I applied late was a big deal. So out of the schools that I got, we made a deal with my family. My family did not want me to even go to Berkeley. That was too far. So we ended up compromising on one of the closest schools to where I live, which was, oh, I forgot, this was really important before that. How did I get to UCLA? Sesame Street is how I got to UCLA. I totally forgot. I'm glad I put Big Bird on there. This is how I learned English, just so everybody knows. The public broadcasting system. My parents only spoke Spanish. We had a rule in my house. From the door in is Spanish, from the door out, you can speak English. If you don't speak Spanish in the house, you don't get fed. So inside was like Spanish. But outdoors was this. However, you know, especially even going to school, I knew English, I learned English from this, but I didn't speak it in my house. So I was really shy to speak it even when I got to school, even though I could, but I was really shy. And what really helped were things like Sesame Street. And the reason why is, if you look at it, I loved it, right? You see people from Puerto Rico, you see people that I think are from New York, you see Big Bird and the Count and Cookie Monster. You can see that they all look different and they were all speaking together. And this helped me feel like I could actually speak with those around me, because that Sesame Street neighborhood did not just look one way, and that really helped me. The other thing that was super important for me to end up going to college, you know, I told you I ended up going with my friends and I took all the right courses and everything. And that was because of really things like middle school. Mathematics to me was really, it kind of blew my mind. And the reason it blew my mind, not necessarily if I'm good or if I'm bad, but you can be absolutely silent. You do not have to speak, yet you can communicate through math. You can write it up on a board and you can put 2 + 2 in any language. Someone will be able to understand the equal sign and put a 4. What's more, once you start getting into more advanced things, right? You can do an equation, you know, y equals x squared. And if you know how to graph it, you'll know that that actually represents a visual. That's actually a graph, right? You can actually get then even a parabolic shape. We don't have to speak the same vocabulary, but you'll be able to understand the language of math. And that blew my mind. I loved it because it was the one class where I didn't have to talk, but I could even work in groups, and that was great. Math is also the foundation of science. Everything is based on it. The physics, the biology, everything has math in it. So that to me started letting me know, okay, there's ways we can communicate differently. That's what led me to UCLA. The reason I went to UCLA is I could walk to the train station, take the train station to the station in downtown. From the station downtown, I could take a bus. From the bus, I could then walk and make it to campus. But I lived in the dorms, which was really important for me. I had that opportunity at the time when I went to college to do that. And to me, that was important. The beginning of college, you take these classes with like 500 people, and you're one of them, and you're a number, and that was both bad and good. But it wasn't until I got to my upper divisions that I found research. And I found research through development. So I'm not really a developmental biologist, but I study aging. I know it doesn't quite make sense, but it makes sense in my head. And I learned about this one experiment, which I thought was crazy. You take a newt is sort of this little, almost like salamander type thing. And you can actually take it when it's fertilized and it starts making that embryo. And you can actually take a piece of another embryo that's even a different color. You can attach it to this developing embryo, and all of a sudden there's information coming and it's communicating with each other so that it can make the Siamese twin. And you can even see some parts are black, some parts are white. They can communicate just by putting one piece of an embryo next to the other. I thought that was fascinating because that meant things communicating. From there, there's amazing programs. Make sure you guys take advantage of them. I had the MARC program, which all of a sudden I didn't have to work 3 jobs anymore. It paid me to do science and research, and I took it. As part of these programs, I actually went to UCSF when I was an undergrad, and I actually did a summer program. This person's lab, Alan Bosman's, where I did my internship, he's now my department chair. So you never know where life will take you. So just make sure you guys always on your best behavior. From there, then I went to Stanford. Again, by the time I went to get my doctoral degree, I had convinced my parents to let me go anywhere I wanted. But they still kind of a little bit guilted me, and I was kind of on the fence anyways about school. So I went to Stanford. It was a great decision. I loved it. Now, you guys saw about the embryo, and I told you, oh my God, the embryo can talk with the other embryo and you can make a Siamese twin. So I kind of used that same idea, but in the context of aging. And what I wanted to know at that point is there ways that our tissues communicate with each other? And I used this really old technique. It was first described in the late 1800s, where you physically sew an old animal with a young animal, and you start asking about blood. Does the young blood influence the old blood influence the young? Short answer is yes. old blood can age you like really fast, but young blood can actually reverse some of that. Now, as part of that identity, right, I'm at Stanford at this point, I'm kind of starting to apply my interest in science. Of course, you have also your personal trajectory. So as at Stanford as a graduate student that I met my partner, now husband, Greg, he's originally from Switzerland, also a scientist, so we kind of geek out sometimes on science. And that all happened sort of during that time. And it was after I sort of met him that, you know, I had that opportunity to kind of go on and start my lab. However, it wasn't by design. I was actually going to go do the postdoctoral training. I was going to go to the East Coast, and then the guy ended up moving, and my family was like, "You're not moving to that particular part of the country." First, because my parents were really scared because they were immigrants for a long, or illegal immigrants for a long time, and I'm part of the LGBTQ plus community, so we decided let's not do it. So then I ended up staying an extra year, and then this opportunity came. So I said, why not? Let's just try and start my lab. I will say always, if there's an opportunity, just walk through the door. What's the worst that can happen? If it fails, you weren't going to do it anyway. So that's sort of been a philosophy I've taken. Because if not, I'm really good at talking myself out. That brings me back to the science, which is, you know, can we preserve our identity by countering aging? I want to remember all of these experiences. I want to remember the good, the bad, what's made me. I want to be able to, you know, tell people, hey, even if you don't think you're going to get there, look at this. I mean, I didn't even apply to college initially. So for me, all of this makes me, and it's part of what makes me a scientist and a professor. So I want to preserve that sense of identity. But that's a big question. Can we preserve identity by countering aging? I'm a scientist, which means I like to reduce things and ask questions and build hypotheses that I can test and try and prove them wrong. I mean, that's what a hypothesis is for. So I had to change my question a little bit. And what we study in the lab is, can the effects of aging on the brain be reversed? So I know that cognition is dependent on cells and molecules and proteins and genes within the brain. So now I want to understand, well, how does aging impact those aspects of aging? Because I know if I'm able to understand that, I'll be able to understand cognition. Now, when we get older, we all know that there's cognitive decline that happens. So if you just look at age around retirement age, so when people are like in their mid-60s, you start noticing cognitive decline. Now, around the same time, we have an increase in the vulnerability that we have for diseases like Alzheimer's disease. So there's this intimate relationship. You're getting older, you're more susceptible to these diseases. So something about aging is happening. Even people with genetic predispositions to Alzheimer's, they're not getting it when they're 20. They're getting it much later. That means something about our body when we're young naturally has the answer because it's been doing it the whole time. It's able to fight off all of these insults, even genetically, when we're younger. That means all we have to do is try to decode it. Can we figure out why? And that's what my lab is doing. Now, there's many ways you can tackle it. We focus specifically on what I call interventions. So I don't necessarily want to drill a hole into your brain and like change your genetics and all of that, because that kind of freaks me out. So I want to try and figure out ways that I can target other parts of your body, never even getting into the brain, but having a huge impact on the brain. Again, that idea of the parabiosis, or that idea of the embryo, there's signals that can talk to each other. So I just want to figure out what those signals are without ever having to get into the brain itself. Now, what do I mean by cognitive decline? Now, in the lab, we're able to study certain types of cognition, memory being one of our main focus. So let me use an example of one of the types of things that we study. So we're in the Bay Area, which means there are so many Teslas, especially doing traffic. It's absolutely absurd. So let's, if this is the same on campus, okay? So let's assume that I'm parking the car and all the other faculty just got their new Teslas. I'm not quite a full professor, so I do not have a Tesla. They're just getting more expensive. It's crazy. So let's assume that I can afford one. I parked my car, okay? And maybe, you know, there's the view of the bridge because I'm up on the hill near the Golden Gate Park. Maybe, you know, June is coming up, so maybe Pride, maybe there's a flag nearby, maybe my favorite food truck. I'm going to come in the morning, park, come out, and then my brain's actually naturally going to look at cues around where I parked. And, you know, I love the view of the bridge, so I always try and kind of park near there. So my brain's going to know, oh, yes, we parked our car close to the view of the bridge, but pretty far from the food because I'm trying to diet a little bit. So my brain just knows this automatically. You go, you come back to work, it guides you naturally. As you get older, this becomes more difficult. And maybe you've seen people in the parking lot with like their car alarms. If they're a little bit on the older side, that is your brain on aging. If you are young, and this is happening to you, you are chronically stressed, because everyone does it. So just keep that in mind. Okay, so these are the types of cognition that we can start testing. And the reason we can test this is, just like we use cues to find our car, animal models also use something similar to find location. So we use the mouse, and the reason we use the mouse is it has some pretty good hallmarks that kind of model aging. Plus it's amazing for, like, you can do genetics, you can try drugs, you can do a lot of stuff to them, and you can still do function. So here I'm showing you a young animal, here I'm showing you an old animal, and I just put kind of on the right-hand side, like, how do they match up? So like a little mouse that's like 3 to 6 months, like someone in their 20s. And a mouse that's closer to like 2 years old is more like someone that just retired. And those are the ages roughly that we use. Now think about it. This guy on the left, he's pretty shiny. He's still black. You see that? Look how much gray hair the old guy has, right? If you were to touch their skin, same thing. Everything, all the hallmarks we get. They're a little more plump. You guys see that, right? They're pretty plump. So that happens with aging too. So we can study all of this. Now, what's interesting is you can study the cognition as well. So just like that Tesla that you can find because you parked near the Golden Gate Bridge view, these little guys, if you put them in a pool of water, they're great swimmers, they hate it. So they're like, I want to get out of here really fast. So they'll find a hidden platform just based on that cue. So rather than the bridge, maybe a triangle. Young animals are really good at this. Just like a human, right? They can find this little hidden platform. An old animal can eventually find it, but just like the individual using a car alarm, because they don't remember, they go in the wrong direction a lot. So this guy basically made 3 mistakes and went in the wrong direction 3 times, whereas a young one would have learned really quickly how to do it. So we can start using these assays to start thinking about memory. So now we know, oh, there's memory decline because you went from 0 errors to 3 errors. But we can also do the opposite. We can start asking, well, what if we take an animal that is already impaired? Can we do something to it to make it look more like the young? And again, this isn't like we're knocking something out or no. This is just natural aging. Most of what we do is in natural aging. I just wanted to show you an example of what this looks like. So on the left is a little young guy, on the right is an old guy. So you'll notice the old guy, like, he can swim. They hate water, so he's really trying. The young guy that you saw, like, he took his time, but he was kind of scanning. And he's like, oh, I remember this. And he already found it. He found the hidden platform. And you can see the little old guy frantically trying to find it. They're perfectly normal swimmers. So remember, this is not hurting them. It's just, yeah, they're just, probably not feeling great. And he found the platform. So you see, they do get to it. Within a minute, they get to it. But you saw how many times they went into the wrong arms. We just quantify that. So we make it quantitative. As biologists, we made it quantitative. Now, this is governed by one part of the brain in both humans and mice, same region of the brain. It's called the hippocampus. I'm showing it to you here in kind of that salmon color. And remember how I started with identity, then we moved into cognition, then I'm studying in part of cognition memory. And that memory is governed by this one region, the hippocampus. Now, within that region, there's changes that are happening that are causing that decline, or normally when we're young, that are preserving cognition. We have our neurons, and our neurons are our main form of communication, and they sort of communicate one to the other. And that happens when they make those physical touches called synapses. So on the top, you'll see that there's almost like a stem of a rose with little thorns. Those are just spines. That's where those contacts happen. Those are the spines. And they become very dysfunctional. A lot of times they go away or they become very weak. So it's not that the neurons are dying when we're old, but those connections are getting weaker. We have regeneration stem cells. We don't have so many of them as we get old, and they're not very good. So we have this decline in regeneration. There's a lot of inflammation. So the immune cells that are in the brain are getting very active and very angry, and they're just going to put a lot of inflammatory molecules all around. So you're going to have more inflammation. And even, the whole brain, right, has all the vasculature. If you think about it, every tissue in your body is exposed to blood. That's how we get nutrients and oxygen. It's exposed to blood. And that happens through the vasculature. So it's really important for the communication. As we get older, the barriers, the vasculature actually changes. Oftentimes it becomes, in some cases weaker, in some cases it just disrupted, like molecules that can get in just change. So my lab studies all of them. Now, the idea is, do you have to like reverse everything in order to restore cognition? Do you only have to do certain ones? Is it a particular combination? So we're trying to figure that out. And then the other aspect of it is that all the cells are still there. You know, once we get into full neurodegeneration, once we get into the state of Alzheimer's, then you have death. And once you have cell death, right, we can't bring those back. We can maybe bring them up and compensate. But when we're older, prior to that, we can still protect it. So during that cognitive decline, mild cognitive impairment, we have a lot of opportunity to do that. So that's what we're doing. Now, I mentioned at Stanford, you know, I used this old model where we connected them together and we realized, wow, like young blood is great and it can actually impact a lot of different like cell types and do it through different factors. We were really excited about that. But you're not going to hook yourself up to a young individual when you're older. There is some craziness happening in the Silicon Valley. I am not a fan of it. So no, you should not take your son who is like whatever, 17 years old and bleed them and put their blood into you. They're actually doing it. This is like stories on like the New York Times. All of us doing this work, we're all like, do not do that. That's not what we're saying. You cannot constantly do blood transfusions. It's not very good for your lungs because of the pressure. Needless to say, there could be infections. So the best approach is for us to identify factors that will have a benefit. Okay, but there are craziness. So you'll hear about this in the news. The other thing I want to point out is a lot of people talk about anti-aging strategies. I don't like that term, okay? Because aging itself is not bad. There's a lot of great things that come with it, right? Experiences. You have like judgment. Some people call it wisdom. There are things that are positive about getting older. So I don't want to stop that, right? All I want to do is make sure that you have youthful function. So that's why I like the word rejuvenation, not anti-aging. And what I mean by rejuvenation is I just want to preserve your cognition. I want to make sure that we maintain our identity. So I don't want to do anything crazy. I don't want to live forever. All I want to do is if you're meant to live 95, 100 years, you're living them well until the end. And then at the end, I just want to shorten that amount of disease, lengthen the health span, how long we can be healthy. So that's what we're doing here. However, in humans, there's a lot of ways that we already know are really good at sort of keeping you healthy. So no, we are not going to show ourselves to young individuals. Some people are doing this, but it is not possible long-term, and there's not enough blood in the world for the people that have Alzheimer's right now. So no, that's also not a tenable approach. We also have interventions, for example, exercise. We know people even with Alzheimer's, if they are physically able to exercise, and it's not crazy amounts of exercise, just some exercise, it's able to slow down the rate of cognitive decline. You do it when you're younger, it can actually prevent some of the cognitive decline from happening. It's really incredible. The problem is as we get older and people with dementia become frail, you cannot physically do the exercise. So we know that there's an intervention that has benefit, yet you're physically unable to do it. So we need to figure out how can we give them that benefit without the actual mechanics. Similar to caloric restriction, we know caloric restriction is great, even in monkeys, like they can live longer. They're angry, but they can live longer. So again, you know, like as you're getting older, you're not gonna not eat because you're an elderly and you're probably frail. But we know there's benefits. So again, can we mimic it? So we're really interested in these mimetics. So I'm gonna give you a little bit of the science that we're doing around exercise. So we and now others, people from Stanford and Harvard and Australia and Europe have now shown that you can actually take the benefits of the exercise and you can actually transfer them. So as you exercise, it's changing your body, and that changing your body is reflected in your blood. You can actually take the plasma and we did it in animals, just so you know, but I use the humans just to give you an idea what's happening. So you can exercise an old individual, take their blood, inject it into an old individual that's never exercised, and you can trick the brain into thinking it just exercise, and it like boosts stem cells and it has a huge impact on the brain. So you don't actually need the mechanics, you just need the signals, and the signals are enough to improve. I will say it did not make them skinny. Everyone always asks me, no, but your brain is like really good. So this we did in the animals. That's how we did it. So we took them, we put them in little running wheels, and you just put the little wheel in the cage and they just like kind of, you know, go for it. They run kilometers every night. We have a Bluetooth on the wheel, so then we can basically track them. So it's almost like a Fitbit for the mountain. So we know exactly how much they ran, we can get the blood, and we do the behavior. So let me show you a little bit. And again, remember that we count number of errors. So, for example, on the wheel here, on the maze here, you'll see that that's 3 errors. So these are all old animals. So they are all old animals. A lot of these are actually siblings. In black, you'll just have animals that just got normal blood from an animal that's like sedentary. We call them like the couch potato mice. And then in blue, we have the animals that have the runner plasma. And you'll see that the number of errors go from like 3 to like 1.5. So not as good as like a 20-year-old, but this would basically be like taking a 70-year-old and making them act more like late 30s, which is, I'm early 40s, so I'm still good. So this is great, but I just told you about like this, the blood is great, but we can't do it for everybody. And we don't think it's tenable. There might be a whole bunch of risks associated with it. So we wanted to start figuring out what is it that's going on in the body. So we took the blood of all these animals that either exercise or didn't exercise, and we just looked at their proteins, like what is changing in the blood. And for that, we did an approach called proteomics. You use this machine, a mass spectrometer, basically like spits out all the proteins that are there, smashes them, and then reconstructs them, and they can tell you like what proteins were there and how much there is in them. I expected to see muscle. I expected bone. Like, I thought it's going to be a whole bunch of signals coming from bone and muscle because you're exercising. And a lot of the factors, most of the factors we got were from the liver. And I was like, the liver, what on earth is going on? Why the liver? A lot of it is metabolism. And I was like, okay, I can see that. That kind of makes sense. And one of them was an interesting molecule that we pursued. It's known as GPLD1. I'll tell you what that is. But I'll tell you why we pursue it. So you get a long list of factors that all could do something. And then what we do is we first validate. So here we took their blood and we just ran the blood in that big square, you're just seeing the blood, and we can separate all the proteins just based on their charge, right? The proteins are either big or small, and they just go down this gel at different speeds, so we can just segregate them. And then we can actually just kind of stain for the protein that we want with animals. And that's what we did. And you can see that the sedentary old mice have a little bit less in that band all the way, that dark black band. You'll see it's a little bit lighter. The runners are darker, so there's more in the runner. And we will only do experiments in mice if we see the same changes in humans. So we had access to the Memory and Aging Center at UCSF with, they've got some people that are over 70 with Fitbits. We were able to track their distance. We collected their blood, and we just asked, are there differences in the same protein in an old individual human, if it's more active or less active? And here we use the same exact measurements, so focus on that dark band, and you'll see the first 4 are a bit lighter, the second 4 are a bit darker, just like the mice. So we know in humans, this same liver protein goes up in your blood with activity, which is great because that gives us sort of a molecule can become therapeutic. You can make a small molecule, you can increase it in your body. So we're like, okay, that's a great candidate. And like I said, this is predominantly expressed in your liver. So your liver is producing. So what is GPLD1? I think of this particular protein, it's an enzyme, and I think about it as a scissor. So it turns out that in our cells, we have certain proteins that kind of hang out on the cell surface, like they're just up there. And the way that they're up there is that they have to be anchored onto that cell surface. And that anchoring happens with a fat tag. It's just literally a little piece of fat that's on the protein that tethers that protein to the cell surface. And what this particular liver enzyme does is it recognizes the fat tag, goes, nope, it cleaves it, and then the protein can be released. And that's what we see here. It comes, it cuts it at that GPI anchor, that fat tag. And then what happens when the proteins are released from the cell surface is they can go on and activate a whole bunch of other proteins and just got a whole bunch of stuff going on in your body. Or they can go on and say, nope, you're going to stop working. It can inhibit or prevent different signaling from happening. Or the protein just goes away. It just gets, kind of degraded and just goes away. So a lot of things can happen. There's also a lot of these proteins on the cell surface. So we have a master regulator coming from the liver that can cleave a lot of proteins and can do a lot of stuff after it cleaves a lot of proteins. So we don't know too much yet, but we know something. So the first thing we want to know is just what happens if we increase this in liver? And we went again into our mice. Now, instead of exercising them, all we did is we used an approach to increase this protein in the liver. Now, we used a kind of a trick as well. We actually know what part of the protein is really important to kind of cut. So we kind of know what makes it into that scissor quality. So we made two types of GPLD. One is the normal one. And one of them, we just mutated it so that little part of the protein goes away. So it can no longer cut. Everything else about the protein is the same though. It's just, it cannot cut. And then we put it into the liver of mice and we did our water maze again. Now in black, you have an old animal that just got like normal control. It doesn't really have like anything in there. In green, you have the mice that were old but got GPLD1. They're really smart. It's like a crazy kind of protein. And then here in gray, just in case you didn't see it, again, in gray, is an old animal that got the GPLD1, it's expressing it, but that cannot cut. So we just took away the cutting ability. All the benefit goes away. That lets us know, oh, you have to be cutting something, and that something must be what's good, what's beneficial. So that's how we start building mechanisms. So now we know this protein goes up in the liver from exercise. We know that that goes into the blood. We know it has to be cutting something on the cell surface, and that leads to benefits. That's a big black box. I love this because these are puzzles that we start kind of figuring out. And I love puzzles. This to me is fantastic. I'm like, okay, let's figure it out. So we had a really good, a really great postdoc who came into the lab and said, I'm going to figure out at least some of it. So he looked and he just said, how many of these proteins are there? And there's about 150 of them. He's like, okay, that's a lot. And then he just used a lot of data sets. You know, we all hear about like, oh, let's get your genome sequenced. Oh, let's go sequence this. Oh, 23andMe. So we're leveraging a lot of those data sets that are out there, a lot of sort of the sequencing data. And what we can do is we just look through the sequence and just says, okay, what cells even have these proteins on the cell surface? And of all the ones that we found, most of them are either on the immune system or they're actually on cells that make up the vasculature. And we thought, that's really interesting, right? Because we're looking at a blood factor. And what would be a great way to communicate with the brain, if not with the vasculature, because that's kind of where the blood factors are going to be anyways. So we thought that's a great candidate, so we went for it. Now, the first thing we did is, again, we only go after things where we have some evidence in human. So we took the same blood samples from those older individuals with Fitbit, and we had MRI for them. And MRI is great, right? Especially for people that have dementia, Alzheimer's, there's these different metrics that we can use to give us ideas about the health of our brain. One of them relates to the quality of your vasculature. It's called white matter hyperintensity. Basically, all it means is you get your MRI, and you're going to have these spots that are more contrasty. And what that means is that your vasculature has these little lesions on them. So a little bit of blood kind of leaks in. So we know that the blood-brain barrier is compromised. What we saw is that the higher the levels in blood of the liver protein, the less of those hyperintensities we had. So that to us suggested, oh, there's a correlation between more liver protein, more blood protein, and better metrics of vasculature. So why don't we look at the vasculature? It seems like a pretty good candidate to pursue. So that's what we did. And again, we leveraged sequencing. Again, so much sequencing is out there from people, from animals, from cells. And all we wanted to know is we wanted to ask, okay, of the ones of these 150, we're going to focus in on the ones only on the vasculature. And then we just ask, have any of them ever been implicated in any vascular function? And we found one. This is another protein. This one's now called ALPL. And this one's interesting because this one hangs out on the vasculature. It's on the cell surface. And what happens is as you get older, more of it accumulates and it totally changes your blood-brain barrier. And we're like, oh, this is too good, because now we're like, we know that there's a target of this liver factor that's regulating the blood-brain barrier. It can even cause like hardening of the vasculature. It's like calcifying. So it's like this player that we know happens with aging. So the first thing we wanted to know is just ask, okay, is it a target? And we use sort of a cell system for this. So remember I told you that if GPLD1 is there, the proteins get cleaved, right? They're no longer attached. And when you have cells in a dish, you have to put media on them. Keep them happy with growth factors, which is, that's how they grow. So that's great, because what we did is in cells, we expressed GPLD1 and ALPL, and we asked, well, if it's cleaving it, we should be able to just pick up the soup, and the protein should be in the soup. And what we did is ALPL has activity. So we looked at the activity. So here, all I'm showing you is cells that express ALPL, this is a control, when we pick up that soup, we detect more of this activity. That means that it got cleaved and it's in the soup. Now, how do I know that for sure? When we express the GPLD1 that cannot cut, look at that. It goes away. We no longer detect it at high levels in sort of that supernate, that soup, the medium. So we know it's getting cleaved, but that's in a dish. What about in an actual living organism? So I hope you guys can kind of see it. I think the next one will probably be a little bit better. This is just expression of the vasculature in red, but I think the contrast here will be better. Yes. So this is just activity. That same way that we measured it in sort of the supername, the soup, we're now just measuring it on the vasculature. So this is the normal old animal, and you can see how dark all of this is on those vasculature. But when we put GPLD1 in the liver, can you guys see that it just goes down? It's getting cleaved, so we no longer detect as much activity. So then we wanted to know, okay, maybe we don't even have to put anything into the liver. Maybe we can start using molecules. So what we did is we made use of a molecule that can inhibit this protein. It doesn't cut it, but it inhibits it. Okay, so less of that activity. And that's what GPLD1 is doing. But we're thinking we want to get closer to like therapeutics. So we actually put that small molecule into the food that the mice eat. So we no longer have to inject anything. There's no like, gene therapy, it's just an inhibitor in the food. And then we just went and did behavior. And again, in gray, you see a normal old animal. Again, it's making about 3 errors. But when we put GPLD1, we know GPLD1 is really good. It goes down to about 1, 1.5. And this is what happens when they eat the inhibitor. And this is only doing it on the vasculature. So targeting something on the vasculature can actually cause improvements. So coming back to this idea, can the benefits of exercise be transferred? We think they can. We're starting to move our research a little bit forward. I really want to get things into people. So right now what we're doing is we're actually trying some of these inhibitors and some of these therapies in monkeys. We have certain monkeys called marmosets that don't live very long. They live, in a lab, they live about 8 to 10 years, but they have the same plex, they have the same cognitive decline, they get Alzheimer's-like qualities. So we're now taking all of our different approaches. We have a few approaches for this. We have the inhibitor, but the inhibitor can work in a lot of places, so that can be a little scary. We've designed also these viruses that target only the vasculature. We're trying to figure out, is that another way that we can kind of do it? And then we're actually using little particles made of fat called lipid nanoparticles. And we're also trying to use that to target the vasculature. And now we're trying those actually in primates because that'll be much closer to the human condition. We're also starting to do some clinical trials, but more on like whole plasma, whole blood plasma, not the molecules. But I think the molecules will be important because that's, you go to the doctor, you get a prescription. They can tell you exactly how much of it to take. It can be regulated. We can go through the FDA. I'm sure companies around, Genentech, all these other ones can start producing it. We can start getting it out there. So that's the hope for us. So coming back to the big idea, can the effects of aging on the brain be reversed? Like Dr. Wise said at the very beginning, we think that we can. I think we have some evidence that suggests that we can. And for me, in long term, it's really about, can we preserve our identity by targeting aging, by countering aging? And I think it's just a different approach. When I first got my job out of graduate school, everyone thought it was a little bit crazy because basically what I'm telling people is you never have to go into the brain to treat neurodegenerative disease. And most of what people have been doing have been trying to target the brain only. So they've been trying to target A beta, they've been trying to target the plaques. When you target these liver factors, it also reduces a lot of things like the plaques. But the way that it's doing it is because it's allowing the plaque substances to go out of the brain and into blood circulation. So we just think we have an alternative approach. And I think it's important to bring in a lot of different perspectives. And I believe that those different perspectives are linked to our different identities and our different experiences, which is why I believe that diversity is so important. All diversity, bringing in different perspectives to tackle problems that are affecting mankind in different and interesting ways, because we never know which one is going to be the one that actually gives us that next therapeutic. And that's sort of how we're thinking about it in my lab. And here's just a picture of everyone that is there, including Ollie. She's very important. She's our emotional support, and as well as my, the postdoc. And I want to highlight also Greg, who is the one that did a lot of the work. So you might have recognized him from earlier. He also works in the lab. Great. With that, I'm happy to take any questions. I don't know how you want to do it.
Yeah. So we're definitely open up to some student questions here and I'll help call on people.
And I'll field some in the chat.
Yeah. we
Also, I should mention, the questions can be on anything. Everything that I shared in the PowerPoint presentation is fair game for questions. I'm an open book, so I'm happy to answer any.
Yeah, Mike.
What was kind of the thing that happened in your life that made you want to pursue this study?
That's a fantastic question. So I kind of skimmed over it, but I mentioned that when I first was growing up, we grew up really poor, right? And we grew up sort of in the middle of gangs and drugs. And I literally remember there were, friends from Columbia across, and they would tell us when we were playing as kids, like, don't eat the sugar packets. And then later I found out, oh my gosh, we were playing with like cocaine. So that's that's my starting point. And then I realized in school, because I did things in school, I didn't have to be home. So, we were quite religious and I didn't love it. So I didn't have to go to church if I was doing something in school. So school for me became almost like a life raft. It kept me from not necessarily doing what I didn't like to do. It gave me some freedom, gave me exposure. I played sports, I did leadership. Like I did a lot of things I normally wouldn't. Like we weren't even allowed to go to prom. So if I was selling tickets at prom, that means I could go to prom. You see where this is going, right? So basically for me, that education was so important. It became part of this identity that I had. So everything about me, now I'm at UCSF, I went to Stanford, I helped my family, I got the nice apartment, like I helped them. That whole thing has followed me. And so much of it was based on that education, science, everything that got me to this point. So for me, one of the scariest things is losing that, because everything so far that has prompted me, kind of got me to that next step in life, whether it be education, financial, everything is linked to kind of the ability to think and come up with different ideas. So I want to protect that. So for me, it's not just about the Alzheimer's, but this disconnect that would happen. That's what motivates me, because it's not just for me. I've met many people, and it doesn't matter what your experiences are, that people with completely opposite experiences, like my husband coming from Switzerland, which is basically, I think, almost like they have lambs there that you can pet, like ridiculous. But again, those experiences are like what's driving him. We have really similar. He's interested in Parkinson's and Alzheimer's. I'm interested in aging. So maintaining that is the reason that I do. Now, why do I take the approaches that I take? That's because of that crazy experiment that someone in college showed me, that you can stick an embryo onto another embryo. I never lost it. It won the Nobel Prize, by the way. And it was also one of the first women, together with her PI, with her mentor, that won that Nobel Prize. That whole idea broke my mind, which is you can stick just a tiny little piece and make a whole organism happen. Yeah. So that combination, is kind of what pushes me.
We have a question on the Zoom. Oh, okay. Yeah, there's many questions. Oh, I don't know if you can speak to this, because I don't know if you've looked at diet, but maybe from other researchers. What are the most promising foods for which this research is being done on that are, I think, what foods are,
Got it.
or what compounds in foods?
Mhm.
Yep.
Maybe that's something that everybody here is interested in, right?
Yeah.
What can we eat?
Yeah. Of course, we're focusing right now on caloric restriction. Okay, so I'll let you in on a little trick, or on a little new thing that isn't published yet, which is a lot of things we know are good for us, like a lot of things. Usually they suck a little bit, like exercise, don't eat as much. We know these are good. Anything, I believe right now, anything that impacts your whole body will have a component, a signal in the brain. Not all of them will work as much as the others. For example, exercise might be really good for regeneration. Caloric restriction seems to be all about antioxidants, so it impacts more like the neurons. But all these different interventions do have a blood component, and they have very different factors that are elicited and induced in your blood. They hit different genes, different cells. But remember, I said we may not have to change every cell. It might be different combinations. And it seems like these different interventions cause different combinations of cells to get better, but in the end, improve your cognition. So we've done stuff on caloric restriction, which is taking away all calories. Now, if we're getting down to the individual components that are there, Yeah, there's some research that's coming out, there's quite a bit of compelling evidence, I think, for like Mediterranean style diets. It's not really about even like eliminating everything, right? I think oftentimes it's just the composition. There's reasons why things like even Atkins will work. The problem is that they're not sustainable or they'll stop working. They like work for a little bit and then end. So the length of the effect of some of these molecules is much shorter, depending on sort of the dietary interventions. People are getting down into like amino acids, right? And like ketone bodies, like they're going into completely different ways of making energy. That, for example, ketone bodies are really great right now for the brain, but they don't seem to be as good, if maybe a little detrimental to some of the bone stuff that's going on, for example, in the context of arthritis. So we have to be careful to understand in what context and why does it work versus not, which is why we like figuring out how things work.
I have two questions.
All right.
Both different. One, you said about experiments with it was a zebrafish because I didn't,
So it's a newt.
newt.
Yeah, it's more like a amphibian.
Okay, how? I'm just going, I got kind of a shock that you said that it surprised you too much to discover that, to know about this experiment that you can put a piece of another individual in that organism and see the outcome. But in class, that has been doing forever.
Mhm.
We improve crops in that way, and we get diversity of flower color by making the inserts.
Mhm.
And so what makes this so different from plants that surprised you so much?
Yeah, so really great question. Okay, there's a couple things, and I'm gonna go back to the plant thing as well. I thought about when I think about sort of the inserts, you make a cut into like, I love the roses especially, right, where you make a cut, you kind of put it in, or you even make like a little window and you can put it in and change sort of the coloring of it. What's interesting about that is that you still maintain the information of the original one and you're influencing it by its environment. But you're not, it's not a cell that can become anything and then turn into something else. In this particular experiment, the cells that were put there had no identity to begin with. It was sort of at the stage of the embryo where there is no neuron, there is no muscle, there's no cue. Like they don't even know what their identity is yet. Yet another cell without an identity can come and tell you you're going to become part of an eye, but I'm going to become part of the eye. We're going to become part of the same exact eye, and we're going to be the same exact cell, but I'm going to be right next to you, but I came from the other one. As opposed to like signaling, and you're going to be like, okay, you're going to form now sort of like this pink rose petal, because a cell that normally influences the petal is going to just influence you differently. This is fundamentally changing almost like it's telling you how to like do the genomics of it. I get blown away equally by like axolotls. It's another weird amphibian. You can cut off its entire arm, right? You can cut off a lot of parts of it and it's that same idea. In an organism, in a vertebrate system, you can actually do that. Now coming back to the plants and how we should be going back to the plants, and I think plants are very important. I do aging, and what are the longest living organisms in the world? We actually have them pretty close by. I think redwoods are crazy. So plants figured out how to live for thousands of years. There are friends of mine in Germany, actually in Cologne, that are now doing work on plants, and they're asking, what are the protective mechanisms in plants? They're figuring it out. They're sticking them with gene editing and CRISPR. They're going into mammalian cells, putting in plant proteins that would never be in a human cell or a mouse cell. They're expressing them. They're making the cells super productive. So is it that someday we might be able to have some like sort of plant hybrid proteins that we put in ourselves, and that's going to just maintain us living a little bit longer? That alone is already happening. So it's a great question in terms of the thinking about it. This is also where I think diversity of thought comes in, because I just don't think there's anyone around until, I think about 2024, and this is the first time earlier this year that someone showed that a plant protein can make a cell function better and live longer in a dish. Maybe someone that had a different life experience, had different knowledge, would have come up with that question a long time ago. So absolutely. I mean, I think it's really simple. The idea, blood impacts the body. Seems pretty simple. Didn't happen until about, 15 years. So I'll probably go to one other person, then we can chat after about the question number.
Is there any specific group that you guys want to like test this on, like someone with like a specific disease? And will you try on yourself?
These are great. Yes, so if you had asked me that question 10 years ago, I would have said, the very first thing I want to try is Alzheimer's disease, number one. Now, I go and I look at clinical trials. And Alzheimer's is one of the hardest ones to test in because you're going to need cognition, MRI. It requires a lot of things. The bar is very high because we don't really have treatments. The way that we're working, though, it's the whole body, right? Basically, we don't just see effects on the brain, we're also seeing effects on the body, on inflammation. So one of the clinical trials right now we have at UCSF is we're applying the same therapies that work on the brain, but we're actually doing it on osteoarthritis in the knee, because a lot of the elderly, it's still not old people, a lot of the elderly, they'll go get knee surgeries, and we know that they're gonna have more osteoarthritis. So right now, we're just putting in some plasma into their knees right after their surgery, and just asking, is young blood better for the inflammation of the knee? Now, if we can show that, we can then start going and saying, now I want to try in this really expensive Alzheimer's trial, because look, I know that it's working at least in old people in another part of the body. So I think about it differently now. Now the way I think about clinical trials is what other indication can I show it before I do the one that I really want?
The question from Zoom from Carla. What are the educational backgrounds or degrees of the people that you work with? I think maybe the people in your lab.
Maybe like majors.
Yeah.
They're thinking about how to get.
Yeah, so I guess it's a huge team, right? Like I showed you right now, the graduate students and the postdocs. But in order to do the research that we do, it requires so many more things. So let me take you through why don't we take yesterday? Let's take yesterday. So who did I interact with yesterday? Yesterday, we had to talk about monkeys and mice. So I met with the veterinarians. I met with the vet nurses as well. I met with some of the engineers because we have to figure out cage logistics. On top of that, I also met with my personal, my administrator, who basically is like a life coach at this point. Dr. Y knows, she basically runs my lab. That's the reason I showed up here at the right time. There's also I have my Propel students, my post-baccalaureate students. So they just graduated from college. One of them graduated from San Jose State, and the other one graduated from Berkeley. I have my PhDs students, so they already have their bachelors, some of them their masters, and now they're working on their doctoral degree. My postdocs would already have their bachelors as well. Let's go through that. So Ariana, who runs the lab, she has her degree from SF State. She has it in public policy and communications, and she's the one that basically runs everything. She also helps run the institute. So she's doing the liaison with all the other faculty, and she's kind of promoting all of that. Obviously, the veterinarian has a vet degree, but the vet techs do not. So those are more trained, so they can actually go to specialized schools. They're the ones that are hands-on with these animals doing this crazy stuff. Then from there, we had the engineers as well. So we had to meet with a mechanical engineer, but we also have structural engineers and electrical engineers, all as part of the medical school. And of course, we have also our finances. So this all has to be funded. So someone has to run all the numbers, and I cannot go near that. That lab would be bankrupt. Yeah, so for that, it's more finance people, economy people. So there's a big breadth. So it just depends on what aspect of the research we're talking about at that moment. But I think I give you a pretty big view.
What about the majors of your undergrad?
Ooh, that varies a lot as well based on their experiences. So I have some that had English and sociology, but then did a master's in biology and then came for their PhDs. People now are doing more computational work. So I have people that have computational or bioinformatics or statisticians that are coming. The vast majority will be some version of biology, physiology, molecular biology, genetics, some version.
Kathleen?
So in research, there's a lot of trials and errors. And have you encountered any errors in the past that you have improved? And this way.
Oh, that's fantastic. Yeah. These papers, the stories that I get to present, I'm probably showing you like 30% of all of the work that went into it, because 70% of the work either didn't work. It gave you negatives, it's like completely like, so I'm showing you like the nice part of it. For example, we tested a bunch of proteins. I have like a list of 20 proteins that don't do anything. Like, no matter where you stick them, how much you put in the blood, it doesn't do anything, right? So I showed you like the cool one that like worked. So that's years of stuff. The other thing that I learned as more as a like mentor, I guess, is the fit is really important. So finding as a trainee, finding the right mentor, and as a mentor, finding the right trainee is really important. Before I thought, oh, you know, I can mentor anyone, anywhere, it doesn't matter. It's good. But it turns out that I have my own personalities, things that work really well. So now when people come, I let them know, okay, this has worked really well historically in the lab. You know, so some personalities will be great and some won't. And it's been really great because sometimes they tell me, oh, my personality, you know, I really like, let's say, someone that tells me kind of like what to do and we can meet every day. And I'm like, I travel a lot, like I'm not around very much. So I'm probably, you're gonna get mad at me a lot. So I can tell them, but you know what, there's a brand new faculty right down the hall, and that's exactly what they're like. And then I make those connections. So for me, understanding that it's about fit, I can have the best intentions that I'd like, but it really is about fit. And explaining to the students, you know, that sometimes it's not that I don't want them, but it's because I think you'll be so much more successful in this other environment. So for me, that was a mistake I made early on. And now I'm just very honest with everyone. And I think about fit a lot more, just as a mentor.
I actually have a question for you.
Okay.
And this is relating to something that I know about you, but one of the
Sure.
personal things you didn't share. I'm aware that during your time at UCLA, you switched majors quite a bit.
That's a great one, yeah.
And here at Foothill College, we have students who are looking to transfer to four-year degrees, right? And they're still trying to figure out what major they're gonna be. But also at Foothill, we have folks who already gotten their bachelor's degree and are coming back to school to change, you know, what they decided they were gonna do in life, right? This happens all the time.
Cool.
And it sounded like it happened quite a bit during your undergrad.
It's not fair. No, that's great. So I changed my major 13 times. Let me take you through them. So I started off as a pure biologist. And then I went and took a big class. I was like, oh my gosh, I'm a number. This sucks. So then I was like, oh, let's go back to my true passion, math, you know, because math is, there's no way math will let me down. I love math. I started going down theoretical math, which involves like, there's like multiple levels of infinity, if you can imagine that. My mom became concerned for me when I was a math major, so I got a minor. So from there, I was like, I'm done with the sciences. So then I became an English literature major. I really liked writing, and it's helped me so much as a scientist because we write a lot. At that point, I'm like, oh, this is great. I can do more. So then I became a double major in English literature and Latin American studies. I found that really difficult, and I realized I don't like history, and it was a lot of history. So then I moved, and I don't know why I left English at that point. Then I wanted to be a classics major. And then from classics, I realized that what I really liked about classics was philosophy. So then I became a philosophy major. And then philosophy made me realize that philosophy has a lot of math in it. So then I went back to math. But then I did not do theoretical math for my mother. And that led me into engineering. So I went to mechanical engineering for a little bit. And I went back to humanities, but I don't remember what I did. Anyway, so I ended up physiological sciences in the end. The reason why is because there was a moment in time when I got that MARC program where I had to stay an extra year. So I was in college for 5 years, not 4. And I stayed an extra year because the program was for 2 years and I was already like ending my junior year. And I thought, okay, that's great. You'll pay for me. I'll stay. So I went to, you know how they have the counselors that like help you with like career. So basically we had my transcript and we had to kind of play Tetris and figuring out like what classes would go for which requirements under which major in order for me to like get out within the normal time. And like physiological sciences accepted all of it. It accepted like my math from engineering. They're like, oh, this will substitute some random like physics course you have to do. And like, yeah, okay, you didn't, technically you need to do like this many history classes, but we'll kind of count your philosophy and classics. That involved a lot of begging. And then, yeah, so it ended up where I was a physiology major and then I had a few minors. So I have like a literature, there's a classics, there's definitely a mathematics.
Awesome. Thank
But
you.
it's minor, so I had the extra time.
A little extra time. So I think a couple takeaways, right? It's okay if it takes a little bit of time to figure out what it is you're trying to do with your life. You can totally take that time and try a bunch of different things out. I think that's a really good takeaway. Another takeaway I got out of it is to talk to your counselor because they'll find you a way out once you do figure out what you're gonna do, right?
Yeah, I have, yes, you're right.
Yeah, and I know that there's still questions. I think, Dr. Villeda is still here for a little bit. We have pizza in the back, so whatever remaining questions you all have, I'd ask you to maybe grab a slice of pizza, make sure Dr. Villeda gets a slice of pizza, and then we can just chat more casually. And then those of you who have to head out, you can head out once you get your pizza.
Dr. Roy, if I have one last.
Oh,
little thing.
absolutely. I'm so sorry.
So I usually like to end with one thing, which is, a lot of us, when we're in our careers or education, think that there's a linear path. We always think that there's a set way to do it, like this next, and then the next thing, and then the next thing. And there is no linear path. So just like Dr. Roy said, I was there for 5 years, finished my major. When I went to graduate school, I actually started in one lab. I got sick about 3 years in. I had to go home for a year, and then I came back and had to completely switch. And I thought, oh my goodness, I'm 4 years in, I'm so far behind everyone that I started with, it's over. And like Dr. Weiss said, in the end, because my postdoc didn't work, I got this other position, I ended up starting a lab. I'm no different than anyone else. So there isn't a linear path, there's your path. So just be comfortable with that. Trust yourself as well, because what your career will look like won't necessarily be the way that someone else's career is. That's it. I'm happy to go hang out and pizza someday.
Can we get that document? And we just had a couple gifts for you, Dr. Villeda.
Oh, wow. Thank you.
Just to remember Phil, we have a t-shirt.
Oh, I want to. Great.
Our new STEM t-shirt. And then two other shirts for
Mine is last post, so I'm here to watch.
Yes. I will be doing that.
Stay for pizza. At this point, we've got a lot of people, so just start with one piece and then we'll see what's left and then we'll go from there. So thanks everybody. Thanks everybody on the day.
I love hearing like